Hi, okay, so this is going to be a review video for the natural selection unit, and I'm going to go into, try to cover kind of all the content, even some of the stuff that we didn't necessarily go through teacher-led in class. So some of the unit work in this unit, we basically had you do the reading, and then we kind of did practice problems applying it. So I want to try to make sure everything that you read about, I'm going to try and cover in this video. So we'll see how long it's going to be. It might be a little bit longer, but hopefully it'll give you kind of a solid kind of review of all the content that both we covered in class and that you read about. So I'm going to start with natural selection, which we did actually do kind of notes on in class. That was one of the only topics we really spent class time just really writing about. And the reason for that is because the whole unit is called natural selection. So obviously that's really the most important thing for you to understand. If you have a really solid understanding of natural selection, then you can handle quite a lot of the questions. Just kind of, they always want to come back to that. It's also a really common thing on the AP test to have a long FRQ, and kind of the last question is, okay, we explain why this would have happened because of natural selection. So it's a thing that they throw into a lot of kind of topics. So it might be all about kind of cell communication and then explain natural selection of this part of it or something. So it's something to know about. So I'm going to do kind of, we did a pathway in class. I'm just going to do it as numbered steps this time, just so you, if you're writing about it, can kind of go through and make sure that you've hit all of those kind of numbered points and an explanation of it. So the first thing, anytime you're talking about natural selection, what makes Darwin's whole theory unique from the scientists that came before him was he was the first one who has said it has to be an inherited trait. So he was really clear that it has to be something that's kind of in your genetic makeup. He didn't understand DNA like we do now, but he did understand it's something that you're born with, that's kind of in your internal programming. So what we know is that there has to be genetic variation in individual phenotypes. So it has to be phenotypic variation, and of course it has to be genetic in nature, which means the variation has to come from DNA differences. So it's not something that's kind of environmentally based. And the general cause of any sort of DNA differences is basically mutation. And at least initially it's mutation. So it doesn't mean every single person is born with a mutation, then things just become lots of variations. But the original source of a new variation always starts with a mutation, or a combination of recombination. But again, initially the reason why there was different options in the first place was mutation. So you can kind of base it all back to essentially at some point a mutation is what caused there to be kind of different genetic variations. So the genetic variation has to exist in the population. Genetic variation itself does not drive evolution. If all of those variations are equally fit for their environment, there will be no evolution. You also have polymorphism. So polymorphism is just many different forms of a trait, and it doesn't have anything really to do with your survival. It's just naturally existing variation. If it's going to actually trigger natural selection, there has to be a threat to environment, threat to survival that favors one variation over another. So you're going to have an environmental threat to survival. Whereby one variation is better suited. And this step here is really essential. So this is kind of the most important step that determines if natural selection is going to occur and in what direction does it occur. And this comes back to the whole emphasis of Darwin's theory was all in the environment. So you can't predict natural selection unless you actually know what the environment is and what's going to happen in that environment. So you have to be able to determine the environmental change and what's going to happen with it in order to be able to trace your natural selection pathway. So the environment determines which variation is better suited, and there has to be some sort of threat that not everyone's going to survive. So one of the examples that comes up a lot is antibiotic resistance. So again, antibiotic resistance starts as a genetic variation in the DNA. So some bacteria just have a genetic trait that causes them to resist antibiotics. It's only an adaptation is only a variation that's beneficial if the environmental threat is antibiotics are taken. So if antibiotics being taken is what introduces a threat, therefore it's what causes that to be favored. At that point, whichever variation is better suited is now called an adaptation. So now we go from having variation to having an adaptation. So an adaptation is now a variation that survives best in that environment. So it kind of links the three together. You have to have an environment where there's variation, then you have to have some sort of threat to survival that favors one. The one that's favored is now an adaptation. And then if you survive longer, then you should be able to reproduce more. And so then we have our last component, which is that survival of the fittest or differential reproductive success. So anything that survives longer has more opportunities to reproduce more. And that's again our survival of the fittest or differential reproductive success. If you're talking on an FRQ, do not just write survival of the fittest. The key, and they always seem to want this exact phrasing, you have to be able to say survive and reproduce. If you don't say reproduce, if you just say they'll survive better and don't link it to the reproduction, then you're not going to get full points. And then if they survive and reproduce more because of the fact that the variation was genetic to begin with, their offspring are going to have the variation as well. So more offspring are born with the adaptation each generation. And that is evolution. So that change over time, that's kind of the overall point, which is evolution, which is my overall outcome of natural selection. So these kind of 4 steps, 5 steps, this is the process of natural selection. And this is the outcome, which is evolution. So again, you just have to be very clear if you're doing an FRQ to make sure you're really clear on the fact that it starts with that genetic variation. It only becomes an adaptation once there's an environmental threat that favors one of them, that causes one variation to survive and reproduce. And then because it's a genetic variation, their offspring are born with it as well. So it's not conceptually very difficult, but just be really clear on those kind of things that you cover everything. A few kind of things to talk about, which are that there's a couple different types of selection. So they kind of occur right here. So this, in natural selection, it has to be about the environment. So what makes kind of Darwin's theory of natural selection unique is that the environment is determining who reproduces more based on their survival. There's kind of 2 variations that we talked about. So one of the things that we talked about is, well, what happens if there's no environmental threat? There's no difference in survival, there's just a difference in mating attraction. So let's say all survive equally, but some attract more mates. So basically the pathway is the same, whereby there is a genetic variation, but instead of it being right here where the environment threatens survival and one variation survives better, it's just that one variation attracts more mates. And so that's going to be called sexual selection instead. So basically, instead of survives best, if it was sexual selection, it'd be one variation attracts more mates. So same kind of basic pathway. It's just going to change that kind of what determines who reproduces more. So that's sexual selection. It's just important to know about because a lot of times it looks very similar to natural selection, but it's truly not. It is its own form of selection. And then the other one is where humans get involved. And in that case, humans favor a variation and cause increased breeding for those that have that variation. So in that case, this is going to be another form of selection called artificial selection. Sometimes called selective breeding. Artificial selection is probably the more proper term for it in an evolutionary sense. So again, in this case, there's no threat to survival, but one variation attracts or is breeded more. Either way, they reproduce more. So it starts with variation, causes one variation to reproduce more, therefore more offspring are born with that actual adaptation or variation. So a similar kind of lineage, just a slight difference in terms of how you get there. The other thing I wanted to go through was a couple of different things about the types. So if it's a trait where there's kind of two options, so let's say there's kind of two phenotypes only. So we kind of have a category, you're this or that. In that case, natural selection is going to favor one, and it often will kind of eliminate the other. And it doesn't necessarily eliminate it altogether. If it's a dominant trait and it's favoring, a lot of times if the heterozygote is equally fit, you'll continue to see rare instances of the less favored trait pop up just because you have those kind of heterozygotes surviving, and then every now and then they're going to produce an offspring that has the less favored trait. So in some cases, it's going to completely eliminate it. In other cases, you're just going to see the favored variation go at really high frequency, and the other ones remain at a much lower frequency. So in this case, you're going to see kind of a pattern whereby there's a clear one takes over. The other option is that there's not two distinct phenotypes, but instead we have what's called like a continuous kind of trait, whereby you're anything on an extreme. So that's going to be anything from kind of short to tall, and you have a kind of what's called a normal distribution, whereby most individuals are in the middle, but you have some on either extreme. So two phenotypes is basically like you're either this or you're this. So you're gonna either have, I'm trying to think of a good one off the top of my head, but you might have type Rhesus positive blood, Rhesus negative blood. Like there's only two options. This or this are the two options of what you can be, whereas a continuous trait, you're anything along a scale. So for continuous traits, there's actually 3 different types. And again, you did this in the reading and we didn't really spend a lot of time on it, but I do just want to briefly touch on it now because there's a few questions that require your understanding of this. You don't necessarily have to have memorized the terms as long as you understand that there's kind of different patterns. So there's stabilizing, directional, and disruptive. So what I'm going to do is I'm going to draw in blue what it looked like before natural selection. So in each case, I'm just drawing, they're supposed to look the same, not the world's best artist. So there's a normal distribution. So for any trait that's continuous, basically meaning it adds up. So essentially there might be 10 different genes. So most continuous traits, the other word you'll see associated with that is polygenic. Because usually the reason it's continuous is maybe 10 different genes control it, like height. Well, just by sheer kind of, if you were to shuffle and look at a statistical thing, most people are going to end up with some tall and some short alleles, and therefore they're going to be medium height. Very rare to get all 10 genes be short, all 10 genes be tall, which is why you're naturally going to have less people at the extremes, just because statistically it favors getting a combination of tall and short. So let's look at what happens with natural selection. So it could be that natural selection actually favors being right there in the average, which is what most people are anyway, and that provides the best survival advantage. So a good example of that is body weight. So body weight, it's better to be average. If you're too thin, you're going to have health problems. If you're too heavy, you're going to have health problems. So individuals who are born at a healthy weight, stay at a healthy weight, tend to survive the best. So that's going to have a curve that looks like this. So we kind of essentially lose the tails, and that middle gets even more pronounced, and we kind of narrow in where you see individuals lie. In other cases, it's going to be better to be one extreme than the other. So if you're looking at like speed, if we're looking at speed in organisms that are running from predators or running towards food sources, it's going to be better to be faster. So in that case, the curve is just going to shift. So you're still going to see a normal distribution. You're still going to see variation. Not everyone's going to be the exact same amount of fastness, but over time, all organisms are becoming faster. So your slower members of your population are still faster than the slower members of the population were previously. So your whole distribution is moving out. And then in disruptive selection, there's an advantage to being either extreme. So it could be like beak shape. It might be okay if you have a really long skinny beak, that's beneficial for accessing this food source. Whereas if you have a short stout beak, that tends to provide better kind of crunching access. I don't know a lot about beaks. I'm just making up those examples. But either one of those is beneficial, but having somewhere in the middle that's kind of neither long nor short doesn't really have any particular food source that's beneficial. And so you tend to see that middle one kind of die out and either kind of extreme. So it's better to be either extreme, but just not in the middle there. So that could be like another example that's commonly seen is like plant height, whereby it's better to be short and have access to soil and water, better to be tall and have access to sun. Being somewhere in the middle tends to not be very well adapted. And a lot of times if you see this, you're going to actually end up seeing speciation, whereby over time they just stop being the same trait. They end up being kind of two different species, one that always has this, one that always have that, and they kind of form speciation, reproductive things that cause them not to reproduce anymore. So when that happens, sometimes those end up just breaking off into two totally different species or two totally different phenotypes. You start to see kind of there's just not a variation anymore. They're either this or this. Okay, so that's just a quick overview of just some patterns. And like I said, the AP test is not going to say which of these is stabilizing, but it may have a prompt and you have to be able to predict which kind of pattern you're going to see based on the prompt. So just understanding that sometimes we favor one, sometimes we change the overall shape of a continuous trait. Those are all ways in which evolution shapes things. Okay, the next thing I want to touch on, and we didn't really go through, like I said, a formal kind of class instruction on this, you just did the scenarios. It's pretty easy, so that's kind of why we didn't spend a lot of class time on it. But I do want to make sure that I do kind of go through and explain it just in case you didn't really follow everything with the scenarios, that you do have a time where it's been directly kind of taught to you. And that's the stuff on speciation and reproductive barriers. So speciation is basically the process by which a group of organisms that had been considered one species stop reproducing with one another and therefore are now separate species. So the key with speciation is they no longer reproduce. And therefore are now considered two species. So the biological definition of a species is all about whether or not they reproduce successfully. It has nothing to do with kind of how much they look alike. It doesn't even really have to do with you would see more genetic similarity, but that's not even how we define it. So you could have two species that are extremely genetically similar, and then you could have another one species that has quite a bit of genetic diversity within its species, and that's not really how we define it. It's just, are they making offspring together? If they're continuing to make offspring together, then we say they're one species. Once they're no longer making offspring together, we consider them to be two species. So then a reproductive barrier is something that causes them not to reproduce. So it's something that prevents successful reproduction. And again, I I think these questions are pretty easy, but I will say that they do sometimes ask kind of specifically about these terms, which is a little bit unusual because a lot of times the AP test doesn't really necessarily care about the vocab, more about the concept. But in this case, they do sometimes kind of use a very specific scenario and then like, which is it? So I would recommend knowing these just because of that tendency for those to show up. So there's 9 different barriers we're going to go through, 6 of which are considered prezygotic. So prezygotic means a zygote is never formed, so something causes the egg and sperm to not successfully fuse. A postzygotic barrier, there is a formation of a zygote, but it either doesn't survive all the way to birth, doesn't survive once it's born, or survives but doesn't successfully reproduce itself. So it's kind of a later on. Generally, postzygotic barriers are more rare. Species generally want to preserve their energy, and having reproductive kind of interactions that don't lead to kind of ongoing success of the species is not in the best interest of the species. So it's rare to see postzygotic barriers. Most organisms that are not going to be successful to mating together don't mate together. So let's go through quickly the barriers that I want you to know. So the first 6 that we're going to talk about are considered prezygotic barriers. So the first one is geographic. So these are all called isolation. So this is going to be geographic isolation. And geographic isolation does have another term that the AP Board would like you to know, and that is also called allopatric speciation. So allopatric speciation is whereby they don't reproduce with one another because they're physically separated. So there's a physical barrier, a lack of interaction. That's why they don't reproduce together. Sympatric speciation means that they are interacting with one another and yet are not successful in reproducing. So the other barriers, all the others would be forms of sympatric speciation, whereby the failure to kind of successfully reproduce is not based on physical location. So geographic isolation basically just means that there is a barrier. So there is a physical block to interacting. So basically they cannot reach each other. So if they cannot physically come into contact with each other, they're not going to mate. So this could basically be like a mountain range. And my examples I'm going to give pretty vague. Separates 2 populations. So I'm not going to give you names of specific species because there's no way you would need to know that, but you might need to be able to give kind of a more generic example that just shows that you know what that means. So this is a prezygotic barrier because if you never get to see each other, you don't make any zygotes together. And so I'm kind of going from most separated to kind of least. So that's kind of the most separated you can get, is that you never come into contact with each other. Similar to geographic isolation is habitat, or sometimes you'll see it as ecological isolation. So it can be called either. To me, habitat isolation just makes a little bit more sense. In this case, there's not a physical barrier in place, but they just reside in different habitats in the same region. So basically they could come into contact with each other, but they just don't. So they happen to kind of hang out in different areas, and so they just don't really interact with one another, but there's nothing physically preventing them. So there's no actual like block, but they choose to reside in different areas. So same basic area, they could interact if they wanted to, but they just don't. So it could be that one species lives near the water and one more inland. So because these guys are staying near the water and these guys are staying all the way over there, they just stop reproducing with one another, even though nothing's physically preventing them from walking between those 2 areas. So that's going to be another prezygotic barrier because they basically don't interact with one another, therefore they don't make any zygotes. The next thing is that they do live in the same kind of region and have some sort of interaction with each other, but their breeding seasons are not consistent. So it's kind of a weird one in human populations because we don't have set breeding seasons, but most plants and animals do. So they don't just breed all the time. They have periods of the year, usually based on kind of when would be the best time for offspring to be born for survival. So they have certain times that they tend to breed. So that's called temporal isolation. And in temporal isolation, they breed at different times of the day or year. So it could be a day, like a circadian kind of thing, one in morning, one at night. Most of the times it's season based, so it's different months of the year. So it could be one species breeds in spring and the other in summer. And a lot of times this actually develops to kind of prevent species that would actually be unsuccessful if they were to try to breed from breeding with each other. So if they're kind of chromosomally complementary, I don't know, I'm trying to think of the right word, unsuited for each other, then what happens is it's not beneficial for them to try to mate and make offspring that aren't going to be successful. So they actually kind of evolve different breeding periods that prevents them from even trying. And that's an overall better outcome if they don't try, if it's not going to be successful anyway. So that's a pre-zygotic barrier. Kind of similar idea about how it's better not to try and mate if you're going to be unsuccessful is behavioral isolation. So if they're not going to be successful in the long run, it's better to not even attempt it. And so one of the things that species evolve to help them know who they should be mating with is specific kind of mating behaviors or rituals that are unique to only their species. So basically, each species has their own kind of rituals that attract mates. And that's usually a good thing because if they only respond to the rituals of their own species, then they're not out trying to mate with someone they're not going to be successful with. So this could be like birds have different mating songs. And so they're only going to respond to a song that's compatible with their own species. So both temporal and behavioral, a lot of times actually kind of evolve through natural selection to kind of favor only successful mating relationships so that organisms aren't wasting their time trying to mate with someone they're not going to be successful with. The next couple are a little bit more rare but do happen. So the next one is called mechanical isolation. And in mechanical isolation, the two organisms actually would attempt to mate, but their reproductive anatomy is incompatible. So they literally cannot form a kind of internal fertilization situation, or if it's a pollination situation, they can't reach where they need to go, so the pollinator's not able to put it there. So there's something that means that the reproductive organs don't actually meet up. So the reproductive organs are incompatible. And so there's a few examples of this, and like there's a snail example, but basically kind of the body structure, we'll say our snail example, do not allow for kind of internal fertilization. So basically kind of the way their shell is shaped, the reproductive parts just can't align because they kind of are blocked by the shell. So that just isn't possible to reproduce even if they were to try. And then the last of our pre, which is actually pretty rare, and you really would only see it and kind of external fertilization. So just a quick recap, the internal fertilization means basically there's some sort of insertion of the sperm into the area where the egg is, and it's kind of one individual with another individual. External fertilization is usually like laying eggs, and then sperm comes nearby, and then they kind of form zygotes outside of the body of either. So that's going to be more common in aquatic species and plants, whereby it's not kind of two individuals forming that zygote in that kind of one-on-one instance. It's more of a kind of put your stuff out there and hope it makes some babies. And so in that case, what you can have is what's called gametic isolation. So in gametic isolation, basically the sperm is just unable to penetrate the egg, even if they're near each other. So the sperm and egg might exist in the same place, but they can't actually penetrate one another. So basically you can see this kind of with aquatic species, whereby the sperm and egg are both in the water, but don't join. And this is kind of a last line to prevent the production of zygotes that aren't going to be effective. So it's kind of like, these two aren't going to work from a chromosomal standpoint. So they perform some sort of kind of barrier around the egg that kind of prevents that from happening. So this is our last that's pre-zygotic, because we're getting pretty close here to a zygote because the sperm and egg are in the same place. So that's our last one that's considered pre-zygotic. The next three are post-zygotic, meaning the egg does get penetrated by the sperm, and yet there's still some sort of kind of further issue that prevents kind of ongoing reproduction as a species. So the first of those is called reduced hybrid viability. Sorry, should have moved that down. So reduced hybrid viability. So viable just means able to survive. So basically they create a zygote, but it never survives all the way to adulthood. And we say to adulthood, most of the time it actually doesn't even survive to birth. So kind of the most common outcome of reduced hybrid viability is going to be miscarriage. And the reason for that is chromosome number. So basically incompatible chromosomes prevent healthy development. So most of the time when you see this, you're going to see a miscarriage. In some cases, the children are born, but they're just weak or not going to survive, frail, have a lot of medical issues, things like that. And so essentially their chromosomes aren't working out right. So if you go back to kind of when we talked about non-disjunction, and humans having even one extra chromosome causes a whole range of issues, most of the time leading to miscarriage. So when you have two species that don't have the same chromosome number, the hybrid offspring has an inconsistent number of chromosomes and usually can't survive well with that. So that's going to be a post-zygotic though, because it does actually have the sperm penetrate the egg. It just has problems after that. Next one is hybrid sterility, which sometimes you'll see phrased as reduced hybrid fertility. Means the same thing. To be sterile is to have reduced fertility. And so in this case, they create a healthy offspring, but the offspring is infertile. And this is one where an example is easy to remember. So the good example of that is a mule. So a donkey plus a horse equals a mule. But a mule has an odd diploid number of chromosomes, which somehow is okay, the combination seems to work for adulthood, but when it goes through meiosis, it can't create healthy gametes. So that's just an example where, therefore, we don't consider those one species because they can't kind of continue their own lineage. So that's post. And a lot of times you'll see this if humans interfere. So another example is like a liger. So if humans are like, hey, wouldn't it be fun if we made you mate and kind of created this cool hybrid offspring? And sometimes we can do that if their chromosomes are similar enough, but they still can't actually go on and reproduce. In the wild, most of the time they wouldn't mate because of behavioral isolation. So in most cases, if their hybrids would be sterile, you tend to see them not mating anyway. They develop kind of a behavioral aversion to kind of prevent that from happening. And the last one is called hybrid breakdown. breakdown is really rare because it's kind of odd, but basically there's a problem in the second generation. So the first generation is both healthy and fertile, but the second generation is not. So they can kind of go for one generation, and then maybe that the second generation is not healthy, it may be that the second generation is not fertile. It kind of differs a little bit. So it's pretty rare. There's a few examples with rice that can do this, but basically offspring's offspring are infertile. So this is where nobody gets to be a grandpa, so they get close, but again, ultimately you're gonna see kind of it die out. And that's another example of a post-zygotic. The other thing that we kind of talked about a little bit in the same lesson as speciation is extinction. There's really just not a lot for me to tell you about extinction. Extinction should be pretty obvious. Extinction is just when a species vanishes. So speciation is one species just splits off. Extinction is they go away. So it just fails to kind of continue to reproduce. And that's usually due to kind of climate change, like mass extinctions are usually due to single kind of huge climate events. Individual speciation events can be due to kind of lack of lack of genetic variability, causing them to kind of not evolve well to their environment. A lot of times it can be related to human influence, sadly. So a lot of extinction events can be related to us hunting, things like that, or kind of small scale environmental changes that they just can't seem to evolve in conjunction with. There's a few questions that are kind of related to extinction, but there's not really any like basic content you have to know. So don't memorize like the 5 extinction events and be ready to talk about it a lot. It's not that important as long as you know what extinction is and just kind of that understanding that a lot of times it seems to be linked to some sort of kind of dramatic environmental event where most species are not able to survive that. Okay. The next thing I want to go over is a couple of kind of evolutionary patterns, which is basically a fancy way of saying 5 vocabulary words, or 6 or so. So there's just a lot of vocab in this unit, which is also why I didn't spend tons of class time just going over it, because it's nothing very hard. It's just making sure you know all these words. So I want to go over convergent and divergent evolution, and then a couple of the other kind of patterns just real briefly. So convergent and divergent evolution, when we talk about these patterns, we're actually talking now about macroevolution. And what I mean by that is we're no longer zooming in on a single organism. So like antibiotic resistance is microevolution. We're looking at this bacteria, this trait, which is antibiotic resistance, and tracking that one trait and that one organism over time. And that's where we're going to end up again, kind of by the end of this video when we talk about Hardy-Weinberg and looking at allele frequencies. But we're going to kind of zoom out for a few minutes now and kind of talk about some of these patterns and evidence and origins of life. So when we zoom out and we're looking at evolution among bunch of different species. So phylogeny, phylogenetic trees are an example of macroevolution, looking at origins, looking at these kind of, several different species, are they becoming more alike or less alike? Those are all kind of macroevolutionary things. We're looking at big picture patterns across a bunch of different organisms. So when you look at kind of several different species, what you can look at is, are those several species evolving to become more alike, or are they evolving to become more different? So not just looking at one species in isolation, but as a kind of group. So if what happens is that over time, they used to be more similar and they're actually becoming less alike, we call that divergent evolution. So they actually become more different over time because they develop, usually because they're living in different environments. One thing that's unique about divergent evolution is that they have a common ancestor, or a recent common ancestor would maybe be the best way of saying it. Kind of tricky when you start talking about common ancestry and macroevolution, because if you go kind of endosymbiotic theory, you can argue we all have a common ancestor. All of our common ancestor is the first eukaryotic cell. So common ancestry is a little bit of a tricky kind of one to put a term to, but we're talking how recent of a common ancestor we're talking about. So in this case, here's kind of my common ancestor, and then over time, as they go into different environments, they're starting to develop differences between them. So it could be that one is moving into the air because it's going to fly, another one's going into the ocean, another one's staying in the jungle, and another one's going on more of just kind of a flat land. And as they do that, each of them is having kind of different needs. And so as their environments start to differ, they're going to start to evolve and change in ways that are better suited to their environment. There's a couple other terms for this. One that you'll see is called adaptive radiation. And the word adaptive just comes back to that whole idea of environment. So it's basically saying, although they started out very similar, as they move out, they're going to adapt over time to those new environments. And so they're going to become more different because of their tendency to need to adapt to a different environmental set of pressures. So adaptive radiation is the same thing as divergent evolution. And divergent evolution, at least initially, causes speciation. So if these were kind of once one species, then as they go out, they're going to become different species. Then it continues to become more divergent evolution. Even once they're kind of already formed different species, those species are becoming more and more unique. And so divergent evolution is not the exact same thing as speciation, but speciation is a form of divergent evolution. It's kind of the first step in divergent evolution, is that they fail to be the same species anymore. And as they move out, what you tend to find is that they started out with kind of a lot of structural similarity because they shared that common ancestor. But as they move into different environments, their structures evolve to kind of carry out different purposes. So those structures that are kind of evidence of divergent evolution are called homologous structures. And homo means same always. So homologous structures are the same underlying structure, but as it's needed in a different environment, it now has a different purpose. So if we look at this example here where I came up with all those different environments, if you look at the idea that they might have all been mammals once upon a time, well, as the kind of ones that stayed on flat land, it might say like a cat paw uses that forelimb as kind of an additional walking one. As humans, we use it as a kind of front hand, and so we use it a little bit differently. The bats evolved it to become a structure for flight, and then whales evolved it as a fin for swimming. It actually has the same bones in all of these. So the same basic bones and around the same placements, but you can see they're using it for dramatically different things, even though they're all mammals. Hold on, we gotta play for a second, sorry.
Which it stands, one nation, under God, indivisible, with liberty and justice for all. And now for the Texas Pledge. Honor the Texas flag. I pledge allegiance to thee, Texas, one state under God, one and indivisible. And now for the moment of silence.
So common ancestry, all mammals, same basic bone structure, but different purpose because of their different environment. Convergent evolution then is just kind of the opposite. So in convergent evolution, they don't evolve together. It's just that the same environment favors the same kind of adaptations. So they start to become similar simply because their environment is favoring the same traits. So in this case, they have no common ancestry. But they become more similar over time. Just due to similar environmental pressures. So this would be like if you were to go to different deserts and you were to go to a desert in Africa, a desert in Arizona, a desert in Australia, you would see a lot of similar adaptations whereby you say, okay, the plants seem to have some ability to store water. They often have some sort of spine or prickle or something that discourages predators from coming and getting access to their water. You might see kind of similar color patterns for camouflage. They might have burrowing to avoid the heat. And so those are just things that help if you're living in the desert. So even though they do it in kind of different areas of the world and they're not evolving together, they're evolving those same kind of features because the environment favors that. So this time they're kind of totally different categories of organisms, but as they come to live in the same environment, which is like a desert environment, I'm not gonna spell it like dessert, you'll see that they start to actually look quite a lot like each other because of that. Not really any specific other terms for that like there is in that case. And so what tends to happen here is that they start to develop similar kind of functional parts that help in that environment. But because they evolve them differently, they don't actually look very similar structurally. So they kind of had to go about getting it a different way. So like, okay, I need some sort of water storage device, and this one evolves it through this kind of mutation, this one evolves it through this mutation, this one evolves it through this mutation. So they get there in different ways. So as a result, the structures don't really look alike internally, but they're now fulfilling the same purpose. So those are called analogous structures. Which are just the opposite of homologous structures. So this time they're different structurally, even though they're carrying out the same purpose. And so an example of this is kind of different types of wings. So if you were to compare a butterfly wing and a bat wing and a bird wing, you would actually find they're very, very different. So bat wings actually have boning all through the wing. Butterflies have no boning. Birds have just kind of a backbone bone and then lots of feathers. And so they look totally different. So totally different bones and materials. But they're all ultimately doing the same thing, which is that they're all providing flight. So they're all going to boost the ability to fly. And of course, these are insects, these are mammals, these are birds, so they don't have that kind of common ancestry group, which is why they evolved them in different ways. Okay, 3 other terms that again are really just straight up vocab, but I do want to just talk about briefly. So one is the term coevolution. And in coevolution, basically 2 species have kind of like a symbiotic relationship. And so because of their relationship, as one evolves, the other evolves in the same way because they rely on one another. So it might be that as flowers grow longer, hummingbirds' beaks grow longer as well. So the idea basically being because of an ongoing relationship between 2 species, as one evolves, the other kind of evolves in response. So basically 2 very different species have some sort of kind of ongoing relationship. So they tend to kind of evolve at the same time. And it can be even, doesn't have to be a beneficial relationship, it can be a predator-prey relationship. So if the prey evolves faster speed, well then the predator is going to have to evolve faster speed to continue to catch the prey. So it basically means the evolution of one species kind of encourages that becomes an environmental change that triggers the evolution of another species. It's basically just still natural selection. It's just that one species is part of the environmental pressures of the other species, and therefore the changes in that species ends up being a driving force for the changes in the other species. So that's coevolution. They're both kind of evolving through natural selection, but because of their kind of reliance upon one another, the evolution of one is going to continue to trigger the evolution of another. The other 2 is kind of gradualism versus punctuated equilibrium. And this is just referring to kind of the pace. So gradualism, which is probably the more common thing, gradualism basically says it happens over time. So let's say this is speed and this is slow versus fast. So this is just implying that basically every generation they get a little bit faster. So over a period of a really long time, you would now be able to look and say, okay, yeah, now they're faster than they were. But it didn't happen like that. It just every generation increased in speed a little bit. And punctuated equilibrium, and this is more likely to happen if there are kind of dramatic events, so dramatic environmental changes like the removal of a certain predator or prey. In this case, it's like this is their speed, whoop, a new mutation or a change in the environment, sudden change. Then it stays the same for a while, another kind of change occurs, and then it increases. So the overall outcome is kind of the same. If you look at it over a long enough period of time, you see a change has occurred. It's just whether or not the change occurred gradually or whether it occurred in kind of a couple of key events. And both of these are patterns that we see. Like I said, this tends to happen more if you have kind of dramatic environmental changes. This tends to happen more over just kind of relatively more stable environment, but just small kind of changes that overall make the species just a little bit better for that environment. Okay, next thing I want to talk about is some of the evidence for evolution. And when I'm saying evidence for evolution, I'm meaning, again, macroevolution. If you're looking at evidence for microevolution, it's a lot easier to do. We actually, I think when you guys were freshmen, we actually did a lab on antibiotic resistance, and we could actually provide evidence of evolution in a 10, 14-day period in a lab. So if you're looking at a single trait, you can do microevolution. There's a lot more ways of supporting that with evidence. But if we're trying to look at a more zoomed out approach, and we are trying to look at like multiple species, kind of more historic evolution, how does it all fit into the big picture, the forms of evidence become a little bit more difficult because we're trying to look at kind of longer periods of time, more species, and so we have to use a few different tools to kind of study that. So I want to go through kind of the 5 things. Again, not something we spend a lot of time in class on because I think it's pretty easy, but do just want to kind of briefly hit on a couple things about those just to make sure that you do know them. So 5 different strategies that are used if we're trying to provide evidence of kind of macroevolution having occurred. And the first one is fossils. So fossils are something often is thought of in terms of evolution. They're not as helpful as you would think because fossils don't really tell us a lot of how. So what a fossil does is say, this is what it used to look like, this is what it looks like now, and you can say, yeah, change has occurred. It doesn't look like it used to. So they provide in kind of the most basic format a way of saying that yes, there's been a change. Sometimes fossils are more helpful than other times. So if you find good kind of transitional forms, then you can actually say, okay, here's some of the like pathway towards change, and it can provide a kind of more complete story if you do have more fossil evidence. So then you can kind of get a little bit more image. The more kind of fossilized evidence you have, it's just not a lot of organisms fossilize well. So it's not the overall greatest strategy in the world, and it's not one that Darwin used. He was not a paleontologist, but it is one that we've looked at since, and particularly when we are trying to answer the long ago questions, fossils are a way of doing that. With fossils, you can have to find the absolute age. So if you want to know absolutely how old is a fossil, you have to do radioactive dating to get an idea for kind of how much carbon is left. So you're looking at like carbon-14 or carbon-12 isotopes and determining how much of it is left in a bone. Then you would have to get a recent bone that's the exact same kind of size and see how much is it, and then you know the half-life. You do not have to know how to do this for the test. Thankfully, that is beyond the scope. So I want you to know that there is a way of working out literally how old a fossil is, and that it involves that kind of carbon radioactive dating and knowing half-lives. You don't have to know how to do it. More likely, you would be asked just about understanding the relative age of a fossil. And of course, the relative age is just an idea of how old or young is it compared to other fossils. So is this the oldest form? Is this in the middle? That can be really important if we're trying to use transitional fossils to kind of tell a story. We want to know which one's the oldest versus the more recent. And you do need to know that in order to determine that, basically the lower down the fossil is, the older it is. So we want to look for kind of where they're located and understanding that the fossils that are lower are going to provide older information for us. The higher the fossils are going to be the more recent kind of fossilized forms. That's really about the extent. I think there might be a couple questions on fossils that you kind of need to know that. Otherwise, they're just a little bit of common sense, like look at the fossil, be able to determine what the change is. Nothing very interesting, exciting about fossils, but they are important for kind of piecing together big picture overall, where has evolution happened over a long enough period of time. The next thing that we look at is what actually Darwin himself used, which is biogeography. And biogeography is exactly what it sounds like. So it's just looking at organisms in their environment and seeing how they look similar or different to organisms in similar or different environments. So it's basically looking at kind of convergent, divergent evolution and showing that if you look at organisms in similar environments, even all over the world, they have similar features. If you look at organisms in different environments, you see the way that they've evolved to their different environments. So this is what Darwin used, and it has a big emphasis on environment, and it kind of shows that idea of what natural selection is saying, that it seems like the pressures of the environment are playing a big role in evolution. So that's biogeography, nothing hugely important to know about it fact-wise, but just do know that that's what Darwin mainly relied on, which is just looking at organisms in their environment and at their adaptations and drawing kind of connections. The next thing you can do is called comparative anatomy. And comparative anatomy, kind of again, just what it sounds like, which is to compare the anatomy of different organisms and look at kind of how similar their structures are. So this is basically looking at what we just talked about. So you're looking for homologous structures, because if you can see evidence of structures that share a lot of bone pattern similarities, that suggests common ancestry, and that suggests divergent evolution. So that provides evidence of that idea that they used to have a common ancestry, but have become more different over time. So looking for homologous structures. The other thing you can look for is analogous structures, which are what provide evidence of convergent evolution, which are things that look very different internally, but are carrying out the same function. Well, that points to the idea that we see the role of the environment and that they're evolving these kind of similar adaptations for their environment. The other thing you can look for is vestigial structures. And a vestigial structure is a structure that's not used anymore. So that's the human appendix. That's a pelvic bone in a whale. That's a femur bone in a snake. And they again point to some sort of kind of common ancestry. The idea being that once upon a time, that must have been used by an ancestor who did need it, and then we just still have it, and we haven't evolved away from having it. A lot of times it's evolved to be smaller, play much less of a significant role, but it tends to argue that why would we have it if it weren't for the fact that at some point an ancestor needed it. So those are kind of the structures you can see. Sometimes you'll see those same terms used not for structures, but for like traits, a homologous trait or a homologous protein. Homologous just means shared among everyone. Analogous means not the same. So anytime you see homologous in that word, that term, it's just meaning different species have the same. So homologous gene, same gene in different species. Homologous protein, same protein. Analogous adaptation, an adaptation that came about separately. So just kind of know that you can use those words kind of a little bit more broadly. They don't always have the word structure attached to them. The fourth thing, which is really similar, but just you can see a little bit more similarity if instead of looking at an adult form, you look at an embryo form. So generally you see more kind of physical similarities if you compare embryos, anatomical, versus if you compare adult forms. And again, that suggests kind of this idea of common ancestry, and also an idea that there might be a kind of shared genetic code. So it suggests that kind of as we start developing, a lot of our early kind of fundamental genes that determine kind of basic formatting and development might be common among species. So that's just what you would use embryos for, is to see a little bit more than you can see just from looking at the adult form of organisms. And then of course the most important, and this is what you're most likely to see questions on, and it kind of goes into what we're going to do next, which is phylogeny, and that is molecular evidence. And with molecular evidence, this is a new form. So this is kind of our newest and arguably our best, which is that we actually look at a DNA basis. So instead of trying to look at like the shoulder blade of an organism and decide, does this shoulder look like this shoulder or more like this shoulder, which ultimately is quite subjective and kind of depends on your own ability to discern differences. If you look at the genes, you can say, okay, this gene versus this gene, which genes are more similar? So it allows us to kind of create an objective indication of similarity. So we can actually kind of, instead of doing it based on what they look like, we should be able to determine who's more related by kind of comparing their genes. So there's kind of two different things you can compare. You can compare the DNA bases themselves, or you can compare the amino acid sequences. And in both cases, you have to use kind of a homologous gene or homologous protein, meaning it has to be something that all the organisms have. So a lot of times they'll use things like there's one of the first enzymes is the first step in glycolysis. A lot of times they'll use that enzyme because we know that all living things do glycolysis. So if I'm wanting to compare across a lot of living organisms, if I use a gene that is known to be in all of them, then I can say, okay, yes, that gene is in every organism. And then I say how similar though is the DNA sequence of it. Cytochrome C, which is used in cellular respiration and the electron transport chain, a lot of times is used with eukaryotes. So we want to generally, hemoglobin is a lot of times if you're looking at all kind of more complex vertebrates. So we just need to make sure it's not something super, super specific. Like you wouldn't want to use the human insulin gene because not all organisms use insulin. And so then you're not going to be able to compare very far back if you're using too specific of a gene. So it just needs to be a protein that you find they all use and use in a fairly similar way. If you use DNA-based amino acids, this is going to be the easier thing to do because it's easier to sequence a protein. So we don't have to use electrophoresis for that. Protein sequencing is a little bit easier. The problem is that if you come back to your genetics, silent mutations code for this same amino acid. And so because of the recurring nature of the genetic code, you can have DNA differences that don't show up as an amino acid difference. So this is going to show you more differences. So if instead of sequencing the amino acid, you actually go to the DNA level, you'll catch some of those third base changes. And so if you're really trying to distinguish between very related species, it'd probably be better to do the DNA bases, because if you do amino acids, the protein might be identical, but if you actually went to the DNA level, you'd pick up a couple silent mutations that separate them. It's just harder to do because you have to use like a sequencing technique. So generally, if you're looking at big differences between species, you just use amino acid differences because they're easier to find. If you want to be more specific, then you're looking at DNA bases. And this should be pretty obvious, but basically the higher the number of differences, the less related they are. Or another way you'll see it done is as percent similarity. So if it's phrased as how percent similar are they, then they're going to be more related. So the main thing I would say on this is just be really careful of, are they giving it to you as differences or similarities? So if they're giving it to you as differences, you're looking for low differences to say they're related. If they're giving it to you as similarity, you're looking for high similarity. Obvious thing to say, but I would just be careful of that on the test to really read carefully what is the information given. Not necessarily hard. There tends to be just like charts that tell you kind of differences or similarities, and you just have to follow those to find the most related. And these kind of go straight into phylogeny. So basically when you use this is often used to then create like a phylogenetic tree. So the phylogeny, and you can use these for phylogeny as well. So phylogeny is just basically taking a group of organisms and determining who's more closely related to who. So it's taking a bunch of species and actually determine the relatedness of different species. So it's kind of the evolutionary history of a group of species. And so you can do that with who has different traits, like whether it's homologous traits or just kind of different adaptations. But of course, the best way to do that is going to be to take your molecular evidence, because again, it's objective and numerical. So if we really want to create kind of a really good phylogenetic tree, we want to take molecular data and use that to guide that. So that's what we're going to talk about next. Okay, before we move on totally from our evidence stage, I do want to just briefly talk about evidence that's related to origins of life. So we actually talked a little bit about this back in biochem, which is a long time ago, I know, and I think we were doing it remotely at the time. So I don't expect you to remember all of that, but you also don't need to actually know this in tons of detail, just kind of more the bigger picture understanding. So what we're looking at is, you know, in terms of evolution, like we said, we were kind of already going macro, so multiple species. Now we're even kind of looking even bigger, like macroevolution in terms of the evolution of life. And there's kind of two things that can come into that. So one of them is going to be the endosymbiotic theory. So the endosymbiotic theory, if you recall, is the theory that all eukaryotes came from the engulfing of prokaryotes. So it's the idea that essentially mitochondria and chloroplasts were once living organisms that got engulfed by a eukaryote and now are organelles. So that's our one that relates to chloroplasts and mitochondria. And so that's an actual theory on evolution because it's trying to explain how we kind of share common ancestry with all eukaryotic organisms. In terms of kind of some of the evidence for that, we kind of talked about it a little bit at the time, but essentially they have their own DNA, which are the only organelles that do that. They self-replicate, and they have double membrane structures, kind of suggesting that, and as they enveloped into the prokaryote, they developed a second membrane kind of around their membrane. So those are some of the evidences that suggest that. Probably the most telling one is the fact they have their own DNA and their own ribosomes. And their ribosomes actually look like the ribosomes of prokaryotes rather than eukaryotes. So there's no reason for an organelle to have its own ribosomes and DNA if it weren't once a cell. So that's kind of one thing in the area of origins of life. And that's a little bit further in because that's assuming we already had prokaryotic organisms, we already had early eukaryotes that were starting to develop nuclear membranes, but now we're just kind of looking at the origins of cell complexity. If we want to go even kind of further out than that, we're looking at the origins of all life. So how do we even get living organisms to begin with? And the argument is that it kind of came from biomolecules. So this is kind of looking at the origin of biomolecules and how then those biomolecules could actually form into an environment that was hospitable for life and form into life forms themselves. So it's kind of way rewind back, and that's why we did cover it during biochemistry. So the main theory that kind of comes up with that one is the Oparin-Haldane hypothesis. And you don't need to know the details of that hypothesis, in part because there are some of their details that were wrong. So they were a little bit wrong about what kind of gases were available and the overall kind of qualities of the atmosphere. But basically what they were saying was that there existed kind of chemical building blocks, even just things like CO2, H2O. So building blocks that had carbon and hydrogen and nitrogen sources. So they said there was quite a lot of methane. We're not sure there's quite as much methane as they thought, but some sort of kind of airborne sources of these. And then what happened is that they kind of were able to, on their own, form the monomers of biomolecules. So we're looking at everything from monosaccharides to amino acids to nucleotides to fatty acids, so that we went from elements to actually forming monomers, and then those monomers were able to kind of independently join together to form polymers such as proteins and DNA. So kind of a weird placement where your course puts this, because it made more sense to me kind of why we talked about it back in biomolecules is when we were talking about monomers forming polymers. That was a good time to kind of talk about how and when that began. But officially the AP course puts it in this unit as part of evolution because it's kind of tying together kind of the origins and history of all life. So that's kind of what the basic hypothesis is just saying, is that the conditions were right. So there was basically the environmental conditions were favorable for this to happen. And so in order to provide evidence of this, there was a famous study done that you, again, don't need to know the details of, but I would like you to know kind of the point of. So the Miller-Urey experiment basically created like a mock environment. So basically it created this big contraption whereby they essentially put in the gases. So they supplied those chemical gases that had the building blocks. They had like a pool of water to represent kind of the vapor that an ocean would give off. And they gave an electric shock to represent forces like lightning. And what they wanted to see is if you put them in that kind of environment that simulated an early Earth atmosphere, would those building blocks form monomers on their own? And they did. So in that environment, they were able to see evidence of amino acids being made from kind of gaseous molecules. So that was the point of that experiment. So basically they input gaseous molecules that had the carbon molecules, it helps I can spell, that had carbon, hydrogen, oxygen, and nitrogen. They had kind of a water vapor source, and they had a simulation of lightning, so an electric shock. And they put it in this kind of closed system. And what they did see was they were able to derive amino acids from that. And they were also able to get some monosaccharides and some carbohydrates and so forth as well. So they were able to show that there does seem to suggest that this is in fact possible. It is possible that the basic environment that we think was there at the time could support the formation of basic chemical molecules, building blocks, the monomers forming. And then from there, those could undergo kind of dehydration synthesis and almost spontaneously create the polymers that would start to then give rise to life. There were some things that were wrong in their kind of experiment in terms of they used gases that we don't actually think now are true, such as using too much methane and stuff like that. You don't need to know that level of evaluation of the experiment, but you need to kind of understand that these two go together. So this experiment was showing evidence of that hypothesis that you can in fact have kind of elements in gas form come together to form those kind of physical monomers that will then become polymers. And then kind of related to this idea, the other one piece I wanted to go through is this whole idea of, okay, well, that's how you would get to the actual polymers, like which polymers came first, like which polymer then gave rise to life. And so one of the hypotheses that I do want you to just know again, fairly briefly about, is the RNA world hypothesis. And the RNA world hypothesis actually states that kind of the first thing that had to develop that then could kind of spiral into everything else was RNA, not DNA. So kind of prior to that, they said, well, DNA codes for proteins. So probably the most important thing was that we make DNA. So if we can use this process to form DNA, then that DNA can essentially be read into making proteins, and then proteins are the building blocks of the living things. So then those proteins build together, and that's how we form a life. So the argument was take the gene first and build from it, rather than taking an organism and then making it a gene. So that was kind of the initial idea, is that we should have genes first. And there's an alternate to that called metabolism first, which argues that kind of actually cellular respiration, things like that, came before genetics. So basically organisms had to have an ability to produce ATP and energy, and then once it got those processes going and the dehydration synthesis, then that could kind of formulate then a DNA basis to then kind of elaborate on that and so forth. It seems like the only one they really want you to know about is the RNA world hypothesis, so I wouldn't worry too much about, like, there's also like the idea that it could have come from a meteor, from space, and things like that. Be comfortable with the RNA world one. So all that is saying is basically that RNA predates DNA. And in that case also predates proteins, true cells, everything. So RNA was the very first one. And there's a few reasons to think that from what you already know about RNA. So it is a simpler molecule because it is single-stranded, and it is what is directly used to make proteins. So if you think about it in terms of even functionality, it kind of has the more important role if we're just trying to expand things. But the other thing that we hadn't really necessarily talked about, but RNA, in addition to being a copy of DNA, which is how we think of it, it also has the ability to act as a catalyst, which basically means it can drive or kind of start or speed up other reactions. So that's kind of a unique thing that we haven't ever really talked about because we've generally said proteins are enzymes, but RNA can actually act as an enzyme. So when they do that, they're called, they have this ribozymes instead of enzymes. So ribozymes are the kind of property whereby RNA actually catalyzes a reaction. So then our argument is that RNA could have actually been the one doing this process. So RNA could have actually existed kind of as the first one, and then it facilitated the production of other monomers turning into polymers, and that kind of is how we form DNA and proteins and then cell parts and so forth. Not a lot of like super important detail you need to know about that. I would just want you to have heard the idea that RNA came first and to kind of understand that as well as some of the other properties, like it can self-replicate, it forms the information for proteins, it's single-stranded. Some of the things you already know, that it makes up ribosomes. So our RNA is how we make ribosomes. So there's some things about RNA that you already know, but the other thing we just never mentioned was that idea that RNA does have its own catabolic properties, or its own ability to catalyze reactions. So that's the other part of that. So I wouldn't get super hung up on the origins of life stuff. All the questions I've seen on it are fairly like just kind of intuitive. So being able to make sense of some information that's in a question. But I did want to spend a little bit of time on it just because it has been a while since we talked about it. And like I said, I think it's kind of a weird placement for it, but you can see questions pop up on it on an evolution progress check. And I think there's a couple questions on the test that are going to show up on it. So just making sure that you're familiar with this basic idea here of this is the theory, this is the experiment that was used to back it up, and when we start talking about which polymers were formed, there's a reason to think that the very first one might have been RNA, and that kind of drove everything else from there. And then endosymbiotic, of course, can show up in both this part and in the cell part. They put it in the course 2 different places. I think endosymbiotic theory is pretty easy, so this is a little bit more complex. Endosymbiotic theory is kind of just your Pac-Man theory, relatively straightforward. Okay, so having kind of gone into evidence, we kind of skipped around a little bit because at the very end, before we moved into the origins of life stuff, we did mention phylogeny. So phylogeny is when we're actually looking at a bunch of different organisms that may be more closely related, or we may be looking at everything from like, tapeworm through to a human. So we could be looking at just kind of primates or a broader group. It kind of can go either way, and trying to kind of show their relationships in terms of some sort of evolutionary relationship. So we're trying to say how closely related are they, how distantly related are they, and so forth. So generally when we talk phylogeny, we talk about using a visual tool. So we want a visual tool that allows us to represent the phylogeny. So we know that in terms of the definition of phylogeny, we're just looking at their evolutionary relatedness. And so now we want to say, okay, well, how do we show that? So there's basically 2 different things that there's a small difference between them. The AP course seems to use them somewhat interchangeably. So that is a cladogram or a phylogenetic tree. So the visual tool that we're going to use is our phylogenetic tree. Or a cladogram. So I'm going to explain we talked a little bit about this in class, but technically there is a slight difference in them, and that a cladogram is linear. So a true cladogram should have kind of a straight line shape to it and should be able to differentiate organisms in a distinct ordering. Phylogenetic trees, on the other hand, are branch-based, and they're usually not as ordered. So they group, but don't necessarily order. I don't want you to get hung up on that definition, and that like if a question says make a cladogram and then to you it looks more like a phylogenetic tree. Again, I think the College Board's a little bit uses the terms somewhat interchangeably, but truly if it's a cladogram, it should have some sort of ordering to it. And so that you should be able to actually tell like most recent and kind of chronological. Phylogenetic tree has less chronology and more grouping space to it. So generally, if we think about a phylogenetic tree, it's going to look more like that. And if we look at a cladogram, it's going to look more like that. So there's basically 2 things that you use to build these. So we have kind of 2 options of how to make them. So the first thing we can use is characters. So we basically use traits. So we're going to see basically which organisms have which kind of traits in common, which organisms have more traits, which organisms have less traits. So we can first of all look at what are called derived characters, which are basically new traits that have evolved. And so when we're doing that, we're basically saying that the more traits that two organisms have in common, the more related they are. So not any sort of rocket science, like pretty straightforward. The other thing is that when you're seeing these tend to lend themselves relatively well to a cladogram because we can use kind of the fewer traits that they have to suggest that those are the older or the simpler, and the more traits they have to suggest they're the more complex. So then down here you would put those that have kind of the fewest traits, and up here you would put these that have the most traits. So then these are going to represent the oldest in terms of evolution, and these are going to represent the most modern or the newest or complex organisms. So if you use traits, you're kind of ordering them in that order. So these here we often call the outlier, and that represents the one that has the least traits. You can do the same here. This would be the outlier here. Same concept is kind of back at the beginning, and then as these branched out, this is kind of where this would be a trait, this would be a trait. So we're looking kind of at traits at different places, but instead of kind of a linear thing, we're just kind of grouping. So all of these ones have this trait, all of these ones have this trait, and they all have this trait. So you can kind of group them that way. So that's kind of a bit, again, probably a more historical way of doing it is if we didn't have molecular data, you can just do it based on who has what. A lot of these times, these questions present with these plus minus charts. And so you're generally just looking through and trying to find the one that, okay, who doesn't have very many traits and putting them as the outlier, and then who shares traits. Okay, if these two have the exact same traits, they would put together. If it's very clear, like this one has one trait, this one has two, this one has three, this one has four, this one has five, that lends itself really nicely to a cladogram for that linear style. If it's more like, hey, these two have 3 traits and these two have a different 3 traits, well, then you're going to look more at a phylogenetic tree because they don't necessarily go directly in an order, they just group together. Generally what I found on the AP questions is they give you a scaffold, so they give you like the picture that they want it to look at, and you just have to plug the organisms in. So that's why it's like I wouldn't get really hung up on that difference because it doesn't seem to matter too much as long as you can make either one that they ask you. And then more importantly than the derived characters, most of the time now we're going to use molecular data. So that's going to be our DNA or our amino acid sequences. And then what you have to do when you do this is you have to be pretty careful because when you're looking at molecular data, you have to look really carefully is did they give you similarities or did they give you differences? Because there's just different ways of presenting it. So sometimes they'll give you it as number of differences. And when you do that, what you're looking for is you're looking at the smaller numbers. So you want to find the smallest numbers. And those that have the smallest numbers are going to be your most related, and you're going to want to put those on branches together. So when you do those, you're basically looking at your small numbers and saying, all right, these two have a really small number, they're a group. These two have a really small number, they're a group. We don't expect the numbers to be 0. We know that these are different species, they're gonna have some differences. And so we're grouping them and being able to kind of group them. A lot of times that works really well with kind of more that tree base, that branching, because we're using small numbers to just say they go together. It's not always as easy to build a complete linear relationship when you have number of differences. The other option you can have is you can have kind of percent similarity. And usually if you have percent similarity, it's like to a model species. So it's to something. So it might be percent similarity to humans, or we're looking at kind of this new species and we're looking at what is the percent similarity towards this. And so if you do that, that usually allows for more of a linear approach because you can go from lowest percent similarity to highest percent similarity. And that allows you to make kind of a nice linear kind of pathway. So there's your lowest, there's your high. So if you get lowest percent similarity to humans, percent similarity to horses, something like that, and it gives it that way to you, that's another option that you can do, is to kind of build from least similar to most similar and do that. Honestly, it's kind of hard to talk about phylogenetic trees just in kind of a summary video. A lot of it's just practice. So hopefully the practice that we did in class will help you kind of be able to work out kind of how to do these and go back to kind of that practice. Again, just in terms of how do you interpret, so just a couple things you need to bear in mind to interpret is that the smaller the branch, the more closely related they are. And hopefully that's pretty straightforward. So the smaller the branch, the more closely related. So obviously if this was like A, B, C, D, and E, A and B are more closely related to each other than A is to C. But A is still more closely related to C than it is to E. So A is most closely related to B, but then A is more closely related to C because they link right here than it is all the way to D. So you're kind of finding these are kind of your nodes. So you want to find where they connect, and kind of the more closely connected they are, the more related they are. So that's probably the main thing you're looking at there. It's just the smaller the branch, the more closely related they are, or the closer they are to each other on a line, the more closely related they are. These are pretty easy to interpret. So if they're physically close to each other, they're going to be closely related. These can be a little bit more fiddly, but you just always have to kind of link back to where they were last connected to one another. One thing to bear in mind that sometimes comes up on questions is that a lot of times in ones like this order doesn't matter. So it doesn't really matter whether you had drawn that as BA or DC. So a lot of times when they're shown like that in a branching, you can't actually differentiate between them unless you kind of have more information, and you can, but the way it's shown here, it would have represented the exact same thing. So that's why it's really all about the grouping when you look at these kind of branch style. So who's grouped together is what's crucial, not necessarily the order that they're presented within those small branches. So there's going to be some more practice ones on the MyAP problems. If you're still stuck on them, come ask me and we can kind of go through them. It's just hard without kind of going through specific examples to really kind of explain them super well, but just kind of make it, that's kind of a little bit of at least the information to remember. You have to know how to do these. We did the in-class practice one, so that's another good thing to go back and open up. And then you did the multiple choice. So if you go back to those, and then you did like 2 different warm-up ones. So I tried to make sure that in a period of a couple lessons, you had looked at some more than 10 different examples of these. So go back to the examples we did, make sure you can follow and understand them. And that's really the best thing you can do, is just look at a bunch and understand how to answer those questions, because you'll start to see the patterns. Like there's only so many different ways they can look. So the more of them you look at, the more familiar you'll be with it. Okay, so we've been talking evidence in phylogeny, which are macroevolution concepts. So this is big picture, bunch of species, how are they all evolving, trying to relate common ancestry. Macroevolution is not my favorite area because there's a lot of extrapolating. So it's a lot of trying to make assumptions from kind of evidence that doesn't tell the whole story. Microevolution is a totally different story. So when we move into microevolution, the key is now we're looking at one population. So not even just one species. We're looking at one species that lives in one area at one time. So super zoomed in. So we can't look at like all goldfish ever. Like goldfish in this stream, if you will. That's who we're looking at. And we're looking at one trait, which is linked to therefore one gene, because one gene determines one trait. So we're looking at a single trait for a single population of organisms. And we're saying, does it change over time? So in other words, do the allele frequencies, because we're talking gene now, change over time? So that's what we're looking at now is alleles. So this is where your genetics is really important. So you have to be really good with your alleles. Like I think I mentioned in class, this is population genetics. So it's really just kind of an extension of genetics, kind of evolution. It's kind of where they meet in the middle. So you have to be really comfortable with the terms of alleles and things like that. So the two kind of, do they change over time? So the two outcomes are yes, no, and then how do we know? So that's kind of what I want to look at for this. I'm not going to spend as long on this only because we've just done it recently. So I tend to feel like there's often a recency effect that whatever we just talked about hopefully is still in your brain pretty well. And we spent a little bit more class time on this than we did some of the other things. So I'm going to kind of go through a little bit more quickly. But if they do the allele frequency change over time, if the answer is no, then we are in what's called genetic equilibrium, which is just staying the same genetically, so that term's pretty obvious, again, for that one population and that one trait. So we're not saying for everything, they might be evolving in other ways, but for that particular trait that we're talking about, they're staying there. So if we're talking blood type in humans, we're saying, okay, there's other ways humans are changing, but for this trait of blood type, we're staying at about the same number of percent have the A allele, the B allele, the O allele. So the alleles are not dramatically shifting over time, we're staying relatively constant. And so in order for that to happen, Hardy and Weinberg outlined some conditions. So we're going to get mighty tired talking about Hardy and Weinberg by the end of this little summary because they were pretty much in charge of this whole microevolution thing. So they said that genetic equilibrium is really hard to do. And so to actually achieve all these 5 conditions long term is pretty difficult. So it's kind of almost more of a theoretical understanding that they need to be approximately maintaining all of these. So of our 5 conditions, the first is it needs to be a large population size. Small populations give too much weight to individuals, therefore chance events have too much of an impact. There needs to be no immigration or emigration, which we're going to shorten by just saying no migration. That's probably the hardest one to maintain, especially if you're looking at like human populations. Mind you, if you're looking at animal populations, they don't board planes and move places. So that's actually a little bit easier to manage in a different kind of setting if we're talking about different species. So nobody's coming, nobody's going. The next thing is that everybody needs to be equally fit. And that can happen a couple different ways. That can be because there's no threat to survival. So if there's literally no threat to survival, then nobody's going to out-survive anybody else. It could be that there are threats to survival, but that trait just doesn't impact survival. So sure, there's things that impact the survival of a cheetah, but none of them have to do with the cheetah's blood type. So we're just saying that for that particular trait, it's not impacting survival in any way. So all equally fit or likely to survive. Related to that, we have to have random mating. So just like everybody's equally likely to survive, everybody has to be equally likely to attract a mate. We talked about this in class, but just remember that doesn't mean for every trait ever. It means for that one trait. So your blood type does not determine whether you get a mate or not. Other things might, but that trait doesn't. And then you need to not have new variations popping up. So if we have new phenotypes showing up in our population, well then that's going to shift my alleles. So basically the kind of development of a new allele, a new version, is going to cause my other alleles to shift in frequency because I've introduced a third option. So we can't have new variations or new alleles, or that is obviously going to change my frequency of my existing ones. It doesn't mean that one mutation can't change what would have been a big A to a little a. It means one mutation can't change a big A into a C. So we can't introduce a whole new kind of allele, or that will start to shift things. So you have to have all 5 of these to be in equilibrium. So this is not a one or the other. If you want to stay in equilibrium, you need to have all 5. If you do not have all 5, then your population will evolve. So if any one of those is not maintained, you're going to instead have evolution. So if the allele frequencies do change over time, then instead of having equilibrium, we have evolution. And so we started the unit by talking about natural selection, but now it's important to realize that natural selection is just one of 5 things really that cause evolution. So it is a way of evolution. It's the most helpful way because natural selection is favoring the environment and making a species more fit. So natural selection is kind of the best way to evolve, if you will, but it's certainly not the only way. So let's look at what are the other ways. So basically, if you do not have a large population size, then what's going to happen is you're going to have evolution, and the evolution is going to be due to what's called genetic drift. And the key with genetic drift is it's just chance. So it's just random chance that's causing some to outsurvive others. So it's not as purposeful. It's not really guiding us towards better fitness to the environment. It's just kind of random chance events knock out certain variations. And it has an overall impact of decreasing genetic variation and not towards betterness, if you will. It just decreases it at random. So genetic drift is not a particularly good genetic force because it decreases our variation and not towards being more fit, just kind of makes there overall less variation because chance events wipe things out. Let's say instead of having a population size issue, you have migration start to occur and individuals start to come into or leave your population. That's also going to cause evolution due to gene flow. Gene flow is actually a better thing. Gene flow tends to create variation, and variation is always a good thing because variation, some of those variations will be beneficial to the environment, so that can kind of drive natural selection as well by having more variation in your population. So gene flow is how human populations evolve because of the rapid kind of movement from one country to another, and we don't have to remain isolated in our populations. And that's creating more variation, which is kind of an overall positive effect. Gene flow in and of itself is not necessarily positive or negative evolution, but by introducing more variation, it has the potential to help a population. Here's the ones we already should know. If all are not equally likely to survive, but instead one of the variations is more fit, then we're going to have survival of the fittest or natural selection. And natural selection is going to overall decrease our variation, but in a favorable way. So natural selection is going to cause an increase generally in the favorable variation. So it's making you better adapted to your environment. So natural selection is kind of the most positive, if you will, of all the evolutionary forces because the end result is better survivability in your environment. If you don't have random mating, but some variations attract more mates, this is where we started this video with, that's going to cause sexual selection. Sexual selection is a little bit odd. It's kind of neither good nor bad. It doesn't really help you in your environment. Therefore, it doesn't necessarily help your species as a whole. It's kind of a little bit of an odd one. So in general, you're going to evolve towards whatever that kind of sexually desirable characteristic is for the species, which is probably overall good for more mating opportunities, but doesn't necessarily help in terms of like resisting extinction and so forth the way natural selection does. And then the last one, which is probably the one that's the hardest to truly maintain, which is that idea that you can't have new variations popping up. Well, of course, this comes back to what we talked about with natural selection. All evolution initially begins with just mutation occurring. So as mutations occur, sometimes those mutations are going to give rise to a new variation, and then that can cause natural selection, just like gene flow can cause natural selection. So anytime we introduce new variations, it can drive natural selection if those variations are positive. So that's kind of, again, the beginning point of all the others is some sort of mutation that can drive the other processes. Okay, so then we're like, how do we determine if that's happening or not? So it's all good and well to understand what it is. I'm just moving my camera around so I have to erase. But then we say, okay, how do we know that? So in order to know that, it comes to this idea here that it's all looking at allele frequencies. So we want to determine allele frequencies and see are they changing or not. So to do that, we basically use the Hardy-Weinberg formulas. And so I'm not going to go into, I am going to review them because I want this to be a complete review, but I did a whole 30-minute homework video on Hardy-Weinberg, so I don't want to spend 30 minutes on it again. You can go back and watch that. I'm hoping that after we've done enough practice stuff in class, you're feeling pretty good about Hardy-Weinberg, so I just want to do the very basics, and then if you want to get more detail on that, you can go back over that. So your Hardy-Weinberg formulas, remember the goal of that is what you have is your phenotypes, because that's the only thing you can observe. But what you want to work out is your allele frequencies. So our goal here is to take phenotype and turn it into allele frequencies. So the test is going to give you the formulas. So you don't need to memorize the P plus Q equals 1, P squared plus Q, 2PQ plus Q squared is 1. That's on your AP formula sheet. That's given to you. I just don't think that's going to especially help you. So what you need to memorize is your steps. So you need to come in on test day knowing the order that you're going to solve these problems in. So that's the most important thing. So you need to know that anytime you're doing a Hardy-Weinberg problem, and I'm going to give you these 6 steps in order. Every now and then the question will give it to you at like step 3 and you don't have to work backwards. Most of the time though, you're going to have to work these steps. So remember, the first thing you want to find is Q squared. And knowing that means nothing if you don't know that Q squared is the frequency of the recessive genotype. And if we say genotype, genotype means 2 letters. So Q squared is little a, little a. And the reason why we can do this is because this is what we can observe from phenotype. Because little a individuals have their own unique phenotype. So we can calculate this by saying the number that have the recessive phenotype over the total number. And again, phenotype is the one thing that is observable. So that is the one thing that we can actually observe. So I can count, I can say, this is who has the recessive phenotype. So that's why we always have to start here, because that's the only thing we can tell from phenotype. The exception, of course, is if it's incomplete dominance, in which case you can tell all of it from phenotype, and then we work those problems differently. You will have one of those are the ones that you can also then go on and do chi-square with. So those work a little bit differently. We did a few examples of those in class. But let's assume we don't have that. Let's assume we have complete dominance. So then step 2 is just kind of a mathematical fact that if we have Q squared, we can find Q, and Q is the recessive allele. So this is not just individuals who are little a. This is anywhere in the population where there is a little a. So yes, these individuals are going to have 2 little a's, and heterozygotes are also going to have a little a. So Q should be bigger than Q squared. That makes sense. There's going to be more little a alleles than there are individuals who are little a, little a. So to work this out, we're just going to take the square root of Q squared. Step 3 then, this is where it does help to know your formula. So the only formula that's really useful is knowing that P plus Q is 1, which is easy to remember and is on your formula sheet. Because P plus Q is 1, if we know Q, we can work out P. So P then represents our dominant allele. Or the big A allele. So again, because they add to 1, it's just going to be 1 minus Q. At that point, you can kind of solve any problem. So I'm going to show it to you in the kind of 6 steps, but do bear in mind that if it asks you for an allele, you actually don't have to do the rest. So on your test, time matters. So if you see the question says, what is the frequency of the dominant allele? Boom, you've already got it. You don't necessarily have to go all the way through to PQ every time. So you can kind of look and see what you need to know. I think the test often asks about heterozygotes because it's fun to make you do all the work. So important to know how to do all 6 steps, but you don't always have to do them. So then once you know P, you can then work out P squared, and P squared is going to be your homozygous dominant genotype. And in fact, I probably could have written here homozygous recessive, just to make that clear. So homozygous dominant genotype is now who has big A and big A. So they're going to have the dominant phenotype, but so are the heterozygotes. So if we don't do this with Hardy-Weinberg, you can't tell who's big A, who's big A, little a from looking. So that's why we have to do this kind of method to determine that. So if we worked out P, this is just P times P, or P squared. So pretty straightforward. And then step 5. Step 5 then is going to be 2PQ. And so that's going to be your heterozygote. And again, heterozygote means 2 letters, so that's going to be a genotype. So that's going to be big A, little a. Like so. And that's just 2 times P times Q. So you do have to remember that is what goes with that. The last step, which is really important, and this is where people miss questions. I just graded my 9th graders, and they were really good at steps 1 through 5, is you have to answer the actual question, which sounds really obvious, but you have to be careful that you're seeing what are they asking you for. So first you need to determine, are they asking you for an allele or a genotype? So you can work out a whole lot of numbers, and then if you're not sure, what are they actually asking about? Does it say dominant allele, or does it say homozygous dominant genotype? So check kind of which of the 5 are they asking you for. Are they asking you homozygous genotype, recessive allele? So check what are they asking for. And then how do they want it represented? So do they want a frequency? And if they want a frequency, then you're going to leave it as a decimal. Do they want a percent? Well, then you're going to multiply it by 100. Do they want a how many? Well, if they want a how many, you're going to multiply it by the population size. So that's kind of your last step, is just to check what are they actually asking me and solve it accordingly. The other thing they can ask you is it an equilibrium or evolution? Well, then you have to do this twice. So in order to really tell over time, you're going to have to do this at 2 different points in time. So you're going to repeat at 2 different time points. And what you'll find is if the alleles are the same over time, so P plus Q at time 1, and you're going to compare that to P plus Q at time 2, and if they are the same, then you're in equilibrium. My flag's getting in my way. If you do that and they are different, then you're in evolution. And that's not to each other, but to 2 different time points. The last part that we talked about was then how do you use chi-square to tell if it's an equilibrium. So then what you can say, okay, is if it's an equilibrium, this is what I know my allele frequencies are. So if it's an equilibrium, even if my population size is different, I should be able to use the Hardy-Weinberg formulas and work out what each genotype would be. And then I can see, is that what I'm actually seeing? Tends to work really well if you do have incomplete dominance, where you can actually observe genotypes. Those are the problems that we've generally seen. There is not going to be an FRQ chi-square problem. I will tell you that. I was going to try and put one on there, but I actually couldn't find an example of it. So it doesn't seem like something they really are commonly asking about. The chi-square problems tend to come more from the genetics kind of unit. I found a couple of multiple choice questions with chi-square, so we did some of those on your quiz. There might be one that's fairly straightforward on the test, but I don't think it's worth like stressing a whole lot about. It's important to keep up your chi-square skills so that it is going to show up at some point on your test, but it's not going to be an FRQ, so don't worry too much about that. I'm not going to go into that now because I just don't want to spend another 10 minutes of your review time on something that is maybe one multiple choice question. But if you do have to do a Hardy chi-square test, you can go back through the video or go over our class examples and kind of see how you would do that. All right, bye. Good luck on your test.